Well, welcome everyone. It's my great privilege to welcome you to Barry's thesis defense. And it was in 2014 that during one of his first rotations, Barry came to meet me and discuss whether he wanted to join our lab. And so I thought I'd surprise you with a little presentation to remind you of what the things were that were keeping us really up at night in 2014. What you might find is that Barack Obama was the president, and America was considered at the height of its diplomacy efforts, and we were just opening relationships with Cuba after a sort of 50 years of isolation. Frozen #1 was the top grossing movie, and it's interesting how Barry has bracketed his entire time with me with Frozen 1 and now Frozen 2. The Winter Olympics was in Russia, so Russia was in the news, and the FIFA World Cup, which was won by Germany, was being held in Brazil. Some of the bad things that were happening at this time: you could not drink the water in Flint, Michigan, because it was full of lead. The Black Lives Matter movement was just getting going after Michael Brown had been shot in Ferguson, and the Malaysian Airlines plane had gone missing over the Pacific. There are some of the top selling songs for 2014, and I think the top one was quite exciting for Barry, Happy. And so we were happy to have him in the lab. Here's a picture of him a couple of years before he came to the lab. I have to say, Barry was not the standard PhD student. So most PhD students come straight out of maybe a master's program or an MD program. Barry came and introduced me to his wife, and told me that he already had a dog. Some of our other students in the lab have spent the first 3 years of their thesis debating whether to get a dog or not. But in fact, Barry had a lovely family, and he was coming to work with us. And I asked Rose, his wife, if I could tell you this story. He was really interested in autoimmune disease, because there was a question at that time about whether she had antiphospholipid syndrome. And so he was really interested in understanding autoimmune disease, and I thought this was quite a romantic reason to get into a certain field for your PhD. Not many people can say I got into this field because of romance. Anyway, this is how we envisioned his PhD, sort of a straight line going up into the heavens of the future. We did start at the bottom. This is one of the first pictures he sent me, and this is the eyepiece of a microscope. And notice that we kind of missed the image altogether. There were some gels along the way that had very little, some ELISAs, and finally we landed up with the data that he's going to show you today. So hopefully you'll agree that there's been some growth over the years that he's been with me. We did also have sad times, and I think Barry will talk about this a little bit after his presentation. But during the time he's been with me, both his parents sadly passed away, and this was difficult times for him. And it's nice that he's worked through that and able to get to us today. We also did have good times, though, and here's all of us. We were one of the top teams at several years' Lupus walks. Here's Barry hiding behind everyone, and you can see his kids over here, and his wife, so we had a lot of fun along the way. But at that point, I'm going to stop, and let's see if I can get this to work. I'm going to hand over to Barry, and he's going to tell you all about the roles of interferon regulatory factor 5 and interleukin-1 receptor-associated kinases in lupus. Thank you, Barry.
Thank you. Thank you for the introduction.
Otherwise, you'll drop it.
Okay, thank you everybody for coming today. So we're going to talk about the roles for interferon regulatory factor 5 and interleukin 1 receptor-associated kinase 4 in systemic lupus erythematosus and the pathogenesis of that disease. Lupus, SLE, systemic lupus erythematosus, we'll use those words interchangeably as we go. It's a complex polygenic autoimmune disease. It's characterized by chronic relapsing and remitting systemic inflammation. One of the hallmarks is pathogenic autoantibody production, which ends up causing damage to multiple organ systems. And some of the facts about lupus: there's over 5 billion cases worldwide. It's estimated it's hard to get good data on it. Case studies have varied. In the US, it's estimated anywhere from 800,000 to 1.5 million Americans. The US does have one of the highest prevalences worldwide. Roughly 90% of the cases are in women. Your risk as a woman, again, the literature varies, but your increased risk of getting lupus is somewhere between 2 and 15 times more than a male. 80% of the cases manifest between the ages of 15 and 44 years old. And along with the end organ damage that it can cause, unfortunately it carries with it an increased risk of miscarriage and preterm birth. And you can see up on the left some of the systems that are affected. It can affect the cardiovascular system. One of the most common causes of complications from it is kidney damage, and that is a phenomenon called lupus nephritis, where immune complexes deposit in the kidney and there's inflammation in the kidney, and eventually, if it's not treated, your kidneys can start to fail. One of the problems with the disease is that there's been a lack of progress in treatment. There's been treatments around that were discovered through trial and error mostly, and they're somewhat effective, but in the last 50 years there's only been one successful new targeted drug. And so we're hoping that if we understand the disease better, we can lead to new advancements that might lead to new and better treatments going forward. So in order to follow the rest of the presentation, there's a couple of genes that I need to talk to you about and some pathway of interest. Because it's a polygenic disease, it's thought that there's a lot of things that contribute. An environmental insult, the production of autoantibodies, and an over-exaggerated response to this. So they've done genome-wide association studies, and they've looked at different genes that are risk factors for the disease. And one of the ones that comes up considerably is toll-like receptor 7. Toll-like receptor 7 is an endosomal pattern recognition receptor. It binds single-stranded RNA, and it provokes an immune response. And it has come up as a risk locus in multiple studies, and we know that enhanced TLR7 signaling has definitely been linked to the increased risk of lupus. TLR7, as you can see, one of the things that it triggers on the bottom left there is interferon response elements, interferon regulatory factor 7 and 5. And interferon regulatory factor 5, which is part of my talk today, has also been associated as a risk for lupus. This is a schematic of the gene for IRF5. It has 9 exons. The first exon has 4 different alternative start points, and there are some mutations in IRF5 that have been associated with an increased risk of developing lupus. The interesting thing is that most of them are not in the protein coding region. They're not protein mutations, they're regulatory mutations. Either at the beginning or the end of the gene, and they have an effect of either increasing the expression level of the mRNA or the stability of the mRNA. So it's suspected that they're ultimately raising the level of the proteins that's around because there's more message around, and that might be a risk factor for the disease. IRF5 we know is triggered by several TLRs, including TLR7. It's a major component of that pathway. And one of the things that we've shown in this lab in previous work is that systemic knockout of IRF5, when you eliminate the IRF5 gene from the entire organism, in our model of lupus, which I'll tell you more about in a moment, it is protective and against them developing the phenotype, the disease phenotype. So that raised some more questions that I will follow up on in a second. But systemic knockout we know is protective, and then they even showed that heterozygous knockout, where you only knock out one of the 2 copies, was protective as well. So all of this fits together into a pattern that maybe IRF5 expression and the amount of IRF5 around is what's tied to disease, or your increased risk of disease. The other part of my talk today, when we get to it, is going to be about interleukin receptor-associated kinase 4. And IRAK4 is a protein kinase, and that means that it has the ability to phosphorylate or add phosphate groups to other proteins, and often that's required to change their shape or to pass along a signal in a pathway to the next step. We know when our study started, we knew that IROC4 was critical to mammalian TLR signaling and responses, and we knew that in humans who were deficient or had a mutation in IROC4 where it didn't work, often as children they were at increased risk of developing diseases and infection, but they could be treated pretty easily with antibiotics or prophylactic antibiotics. And once they reached adulthood, that kind of part of the phenotype went away. So this has been between that and the fact that pharmaceutical companies have had a lot of great success targeting kinases with drugs in cancer and some other conditions. Those two things have made it an interesting target here because people have said, well, if this is something you can live without as an adult and it's involved in these immune system and autoimmune disorders and it's a kinase that we can target, maybe we should look at this. So what we wanted to do was find out more about its role in SLE pathogenesis because it had been looked at in other autoimmune diseases at the time, but not really in SLE yet when we got started. And we also wanted to know if there was any direct evidence for it playing a role as a kinase with IRF5, because we know IRF5 has to be phosphorylated before it's activated. IRF5 as a transcription factor, it starts out once it produces a protein in the cytoplasm, and it's in a closed conformational shape, and it has to be phosphorylated. When it's phosphorylated, it opens up, it dimerizes, it translocates to the nucleus, and then it starts transcription of other immune factors. So one of our thoughts was, well, hey, maybe IROC4 kinase, since it's in the TLR pathways, and we know IRF5 is in the TLR pathways, maybe it could be one of the things that's responsible for phosphorylating IRF5 or somewhere upstream in that pathway. And if so, wouldn't that be a great potential target? So the last part of the intro, before we get to the data, is our model. So we use a mouse model to study lupus. And mice are great to work with. They have relatively short generational times. They're similar enough to mammalian genetics or similar enough to ours. Typically, mice, the B6 mice, which is the main strain that we work with, does not develop autoimmune disease or lupus. But if you make some gene modifications to it, you can get it to have a lupus phenotype that's very similar to humans. The particular model that we look at is called the Fc gamma R2b knockout. So Fc gamma R2b double negative, that's the minus minus. YAA model. And the Fc gamma R2b is an immune receptor, and it acts as a brake. It's an inhibitory receptor. One of the places that it's very expressed is in B cells. It's conserved between mice and humans. And so what you can see in the schematic here, there's a regular Fc gamma receptor on the left. And when an antigen encounters antibodies and forms an immune complex, it'll bind, the antibody part of it will bind with the Fc gamma R receptor and trigger an immune response. That's a high affinity receptor. It doesn't take many of them to do that. When the amount of immune complexes circulating gets high enough, the Fc gamma R2b is a low affinity receptor. So when it gets high enough in concentration, it'll trigger that one to say, hey, we have enough antibodies out there, shut down production. So by knocking the Fc gamma R2b receptor out, we take that break off. And production of the antibodies continues, and it also, for some reason, increases the chances of autoantibodies being, probably because it plays some role in tolerance. I don't know if the literature's been clear on that, but it increases your chances of developing pathogenic autoantibodies as well. So we've knocked that out in our mice, and because of that, they get dysregulation of their B cells and runaway autoantibodies and antibody production. And then the other part of the model is the YAA section. So the YAA is another mutation. Stands for the Y chromosome linked autoimmune accelerator. This was a piece of the X chromosome that spontaneously transferred to the Y chromosome in another line of mice, and initially wasn't noticed. What happened was when it started getting into lines of mice that had autoimmune phenotypes, their autoimmune phenotypes were much worse. And so instead of eradicating it, people said, hey, wait, this is interesting. There's something in this translocation that's making autoimmune diseases worse. Let's breed it into our autoimmune disease models on purpose, and we can study it. So it's been bred into a number of different lines. Ours, it's in the Fc gamma R2b knockout line, YAA, and then what it ends up doing, because it's genes that are normally on the X, in females with X-axis inactivation, it's believed that those genes are silenced on the second copy. But when you put it on the Y chromosome in a male, they now have double expression of these genes because they're on the X chromosome and they're on the Y chromosome, which is not silenced in a male, and they get upregulated expression of the genes in this area. One of the genes that's in this area is TLR7, which is interesting because it ties back to TLR7 being one of our risks and upregulated TLR7 being one of the risk factors. So there's been a lot of looking into whether or not that could be playing part of the increased risk of the YAA section. But we combined it with our mice, And so we have Fc gamma R2b YAA mice that develop a lupus-like phenotype. It's spontaneous around 4 months of age in the males. These are, when I say it's going to be, if it's a YAA, it's the males, because only males have the Y chromosome. So we're looking at the male mice. They get the spontaneous lupus around 4 months, usually mortality by 6 months. They get high antinuclear autoantibodies, which is typical of lupus. They develop enlarged spleens and enlarged lymph nodes. And for us, one of the reasons we use this model also is because they get great, it's a little stochastic in if they develop it, but they do have a very good incidence of kidney disease that mirrors the lupus nephritis phenotype. So it's one of the better models for studying the impact on the kidneys, and our research was in the nephrology department, so that was an added incentive for us. So just to recap before we go into the aims, we identified some gaps in the knowledge when we started. We knew that double knockout of IRF5 was protective in this model. So if you have the Fc gamma R2b YAA mice and you knock out IRF5 on both copies, they don't develop the phenotype I just talked about. We knew that even heterozygous knockout was protective. There had been no successful in vivo studies involving IRF5 overexpression, because when they tried to overexpress, they did some in vitro studies that worked, but when they tried to overexpress it systemically, when you overexpress it, it turns out IRF5 is in some other tissues besides the immune system. And one of them is the liver. And when you overexpress it in the liver, you get liver apoptosis and death. So that's been a struggle with overexpression models in vivo. And so we thought if we could come up with a way to overcome that, that would be a plus. And as I just mentioned, we knew that there's literature out there that Fc gamma R2b and IRF5 are both active in cells other than immune cells. So I'm going to use the terms hematopoietic, and hematopoietic means the cells that come from the bone marrow and from the blood. And when I say that, I'm referring to the blood cells and the lymphocytes and all the immune cells. And Fc gamma R2b and IRF5 are both expressed in all those cells. But there's papers that say Fc gamma R2b is also very active in the liver. And there were some people who have speculated that there might be dendritic cells or other scavenger cells in the liver that could be contributing. And we know that IRF5 is also active, and one of our prior researchers in our lab showed it was in liver, muscle, and adipose as well. And it may have part of a metabolic phenotype. And we were especially concerned about the adipocytes in the adipose, because there's been other studies showing that fat cells in the adipocytes can cause inflammation and immune syndromes as well. So that was one of the things we wanted to pin down, was we wanted to make sure that the Fc gamma R2b disease effect and the IRF5 knockout protection effect that we were seeing in our model were from the immune cells and from the blood, and not from their roles in the other parts of the body. And then, as I mentioned with ROC4, they had shown at the time we started that it was protective in a couple of other diseases, but it really hadn't been looked at in SLE yet. And it hadn't been directly looked at the time whether it was a regulator of IRF5. So our central hypothesis to start out was we were going to hypothesize that both ROC4's kinase activity and IRF5's activation in hematopoietic cells were required for the development of SLE. To do that, we set out with 3 aims. We broke it into 3 parts. The first part was to, as I said, determine if the Fc gamma R2b phenotype disease and if the IRF5 protection was coming from specifically the immune cells and not the other cells in the body. For the second part, we then wanted to determine if transgene expression of IRF5 could rescue the phenotype. And I'll explain there were reasons why we were interested in that. When I get to aim 2, I'll explain what those were. But we wanted to make sure that if we took our knockouts and we put IRF5 back into it, we would get the disease back. And then in the third part, that's when we looked at our ROC4 and its kinase activity. And we wanted to know if it was required for lupus and if it was required for IRF5 activation. So looking at the first part, we decided that to study the effect of where these 2 genes matter, Fc gamma R2b and IRF5, we would create mouse bone marrow chimeras. And this is an interesting technique that allows you to have a hybrid animal. You take a recipient mouse who has whatever genome you want. So in our case, we took a healthy V6 mouse with no mutations that does not develop a phenotype. You expose that mouse to a lethal amount of radiation, and it destroys any of the rapidly dividing cells, which includes the bone marrow and the stem cells in the bone marrow, and you wipe out its native bone marrow. If you do nothing else, that mouse will die within 3 weeks, 3 or 4 weeks at most, sometimes sooner. But then, so that it doesn't die, you take marrow, which contains stem cells, from another organism, another animal, another mouse, and you inject it into them, and the stem cells from that mouse go and they repopulate the marrow and recreate a new immune system in that mouse and rescue them. This is something you might be familiar with people. They do bone marrow transplants and things like that, sometimes with cancer. It's a similar idea to that, except here we're manipulating the system, and what we're doing is we're taking a mouse that doesn't get disease and has IRF5 in all of its tissues, in the liver, the muscle, and fat, knocking out its blood system, its hematopoietic system, and then putting in marrow from, we had two different groups. In the first group, we put in marrow from Fc gamma R2b YAA mice that developed disease, and we let that repopulate their immune system to see if they would get the disease. And in the second group, we took marrow from an Fc gamma R2b YAA line that was crossed with a knockout for IRF5. So we put that into the other group, and so our hypothesis there is that if we were correct, and this is all being driven by the immune system, that first group should get disease and the second group should be protected. If we saw something different, then we'd know that maybe there were other cells contributing besides the immune cells. The first thing that we wanted to check, you want to make sure that it worked when you make a chimera like this. And the way we went about this, there's a marker on immune cells called CD45. All of the mice in our colony have a variant of this marker called CD45.2. The mice that we got as recipients have a marker called CD45.1. It's slightly different. It doesn't affect them functionally. Otherwise, these mice are from the same background strain. So by doing it this way, when we irradiate that mouse and then we put in our mouse from the marrow from our donor, when you go and you check later, you can look for this CD45.1 or CD45.2 to see where the cell that you're looking at came from. If you're looking at a cell in the blood and it still has a CD45.1 marker, it belongs to the original mouse. It didn't die during irradiation. If it's got a CD45.2 marker on it, it came from your donor. So these are the two groups, shorthand IRF5 wild type and IRF5 knockout. These are all on that Fc gamma R2b YAA background. But one got the IRF5 wild type marrow, the other one got the IRF5 knockout marrow. And here we're looking at about 15 weeks after rescue. And this is the CD45.2 donor access, and these cells are all from the donor. And this is the CD45.1 recipient access, and these are the cells from the recipient. And you can see that in both groups, we got about 90% of the cells at 15 weeks were from the donor mouse. So that's an excellent result. At 15 weeks, 90% of their blood system and immune system was from the donors like we wanted. There were 90% chimeras. That percentage continues to climb because there are some B cells and T cells that are radiation resistant or live in the tissue, and they die over time with age. So the further out you go, that'll start to tilt a little bit more, and you get an even better engraftment, but 90% was fantastic as far as we were concerned at this point. So then we started upon death. So we had set a death, because our mice usually die at 6 months spontaneously, we had set an end date for the experiment of 6 months. Unless a mouse became critically ill before then. If we were notified before then, we would have to euthanize it. We would go and get it and euthanize it then, but anything that made it to 6 months was euthanized at that point. And we started comparing markers of the phenotype for lupus. And you can see that in the group that got the wild type marrow, we had spleens that were elevated in weight, and in the group that got the protective marrow, 100's about right. 90 to 100 is about a normal healthy spleen. So we had a strong protection effect when it came to the size of the spleen and the enlarged spleen. We saw the same thing in the lymph nodes. There's some variation in the disease and from mouse to mouse, but all of these are abnormally high lymph node sizes. And over here in our protected knockout, IRF5 knockout group, we got a nice tight group of normal sized lymph nodes. One of the other things we looked at was these anti-nuclear antibodies that I've mentioned. The way we look at that is you have a slide, and the slide has got cells that are sliced so that the nuclei are exposed. And you take sera from the target that you want to examine, blood sera, and you put it on. And because the nuclei are exposed, if there are autoantibodies or anti-nuclear antibodies in the sera that you put on, they will bind to the nuclear proteins on the slide. Then you wash off the excess, and only the stuff that's bound stays. And then you put on a fluorescent detection antibody for antibodies. And if it lights up, you know that you had antibody deposition, okay? So then you know there were autoantibodies in your original sera. The way that you score it is you then, when you get a positive at the lowest dilution that you try, you then dilute the sera out and continue to test. And if it's positive at the next dilution, you keep going until you get to a dilution where it's no longer positive, and you record that as your highest score. And that gives you an indication of the relative concentration of the antibodies, the amount in your original sample. This is what it looks like. Just to give you an example, when you run the test, this was a positive control. You can see that nice light up green. This was one of our IR5 wild type groups, and we got a nice green response, and in our knockout we got nothing. So things are going well. Putting this in graphical form, when we did the titers out, 1 to 30,000 is a very high titer, and some of these mice at the end were 6 out of 1, 2, 3, 4, 6, 7 out of 8 were up at the top range of 30,000 in the wild type group, and they were undetectable in all the IR5 knockouts. So that's as close to a perfect result as you can get, I think, for this. Just a note on the side, you'll see it matters more in the next group, but we also wanted to compare this 2 ways. We wanted to compare it in total antibody ratio level, but we also wanted to look at clinical significance, because people walk around with autoantibodies at the 1 to 100 range and never develop disease. And so we knew that was a possibility, that if some mice scored 1 to 100, they may not actually be sick. So for this score, what we said is, okay, you had to be over 1 to 100 to be a positive. And we also wanted to look at them that way. So in this case, everybody was over 1 to 100 in the wild type group, and nobody was in the knockout group. So looking at renal histology, these are slices of a kidney from these mice. And the top one is a 40x image, and you can see the one on the left. These are H&E stained, and the blue are immune cells. And you can already see from the higher zoomed out that this is a little bit, I don't know with the lights in here, this is a little bit bluer tinged than that one. But then when you zoom in and you look at glomeruli, the actual functional unit of the kidney, you can see it's much more blue. All these little blue cells are immune cells infiltrating compared to the healthy kidney. And you also get, this is actually necrosed. It's a glomeruli that has died from immune infiltration. And we have a crescent forming down here. These are evidences of immune cell infiltration into the kidney, pretty bad kidney disease. And compared to over here in the knockout group, you've got 2 nice, very healthy glomeruli. So again, things are going swimmingly at this point. We checked some of the mice just to make sure that what we thought was driving it was driving it. And we did find C3 and IgG deposition in the glomeruli. So that's consistent with the lupus nephritis phenotype as well. And then an investigator, which was Ramon, who was blinded, I gave him all the slides and he didn't know which group they came from, and he scored them in terms of their kidney damage. So this scale runs from 0 to 4, and you can see on this scale, everybody is up here at a 3, which is necrosis. In the wild type group, there was widespread kidney necrosis, and in the knockout group, everybody, 0.5 is trivial. You can get that with age. So almost no kidney disease in the knockout group. If there was any, it could have been an effect of the radiation or age, but it was so minimal, and there's obviously a huge and statistically significant difference. You also score them in terms of their overall necrosis and crescent percentages, severe disease, and as you would guess from the previous one, that also is the same. Other than this one note, you had everybody up here with severe, a very high percentage of crescents and necrosis, 75% of the glomeruli versus almost none in the knockouts. So all in all, we found the data in AIM-1 to be very supportive of our hypothesis in our first aim. And if you remember, our first aim was to determine whether it was the hematopoietic cells driving pathogenesis and protection. And I think we decided that the data said that yes. It is still possible that the other cells contribute somewhat to either front, in either direction. But from the strength of this data, you would say that the majority of what's driving pathogenesis and protection is coming from the immune system. So from there, that enabled us to move to our next step, which was to try to determine if rescue of the IR5 would restore the phenotype. This was necessary for a couple of reasons. One of them is because, to learn, just tell you a story from example. In our lab before me, when they were first working with systemic IR5 double knockouts, they had a nice phenotype, and all of a sudden they're going along and things are fine, and somebody publishes a paper that says there's another mutation in the IR5 knockout line nobody knew about. We just found it in this other gene called DOC2, and it might be an immune regulator also. So everybody had to stop and look, and then they found out, yes, actually DOC2 is mutated in this line, and it is another immune regulator. So now it threw all the results for the IR5 knockouts into question. It was like, was it really the IR5 knockout that was protecting this, or was some of the protection coming from the DOC2? So they had to go and they had to breed the DOC2 mutation out, and then they had to repeat their studies with the IR5 knockouts to see if the results changed. Most of the results stayed the same. There was a minor role for, I think, DOC2 in some of the B-cell phenotype, which Dr. Yasuda wrote a paper on. And it ended up being that most of the protection was coming from the IR5 knockout after all. But that kind of thing can really complicate you when you find out later, when you haven't sequenced the whole genome and you don't know about a confounding mutation. So there's always a chance that another mutation has arisen in your line spontaneously. And there's also a chance of a linked gene, a gene that would be a gene next to IRF5 that statistically your odds of recombination are very low there, could have come through with the IRF5 gene that could be changing your phenotype. So by restoring IRF5 to these mice, we wanted to prove that it was actually the IRF5 knockout that was driving the phenotype. And if we know if it was IRF5 knockout that was protecting them, then putting IRF5 back should make them sick. So in order to do this, we started with the same model we used in AIM-1. We took bone marrow from those mice that we were using as donors. We had the same recipients, CD45-1, they don't get disease. Mostly we worked with the Fc gamma R2b IRF5 knockout mice as our background for the donor. And then we would harvest from that marrow. Instead of rescuing these mice with whole marrow, we would take that marrow, we would go to fluorescent activated cell sorting, or FACS, and we would harvest just the stem cells from the marrow, bring the stem cells back to the lab, and we would I'll get to the next slide, we would transduce them with a construct from there. But before I do that, let me show you what that looks like. The way we would get the stem cells, we would incubate the marrow with a dye called Hirsch, and all the cells take up the Hirsch at a certain temperature and time, but stem cells are especially good at pumping it out and getting rid of it. So when you run the protocol correctly, and then you bring it to the FACS in the time that's indicated, you end up with a double negative population that has gotten rid of the dye in the channels that Hirsch lights up on, and that double negative population is your stem cells, as long as you're in the right window, because they'll be the only ones that have pumped out the dye at that point. So what we would do is then gate for those and select the double negatives, and this is the marrow, the rest of the marrow would just be discarded. So we brought the stem cells back to our lab, and we would incubate them with virus-like particles generated from a gene construct. We don't have time to go into the whole system, because I still have a lot more data to show. But the idea here is that we're using a virus that is similar to, or based off of retrovirus similar to HIV, to get inside the cells and deliver transgenes that we want to deliver and modify the genome of the target. So you always want a control, and your control is going to be your, we call it the empty vector. So this is a delivery system, and in our case, this delivery system was just carrying two fluorescent marker proteins, a red and a green. And that's your control for showing that stem cell isolation and the transduction process and the integration of the transgene didn't do anything to your phenotype to change it. The fluorescent marker proteins are there so that when you go and you look at your cells later, you can see how effective your transduction was, what percentage of the cells actually picked up the genes that you were trying to insert. In our active experimental construct, we replaced this DS-red section of this construct with a version of the IRF5 gene. And it was an exonal version of the gene, the introns were out. And we replaced it in there, and so the way that this works is a promoter upstream. EF1A is the promoter upstream, if anyone's interested. That would go through and promote our gene that we put there, either DS-red in the empty vector or one of the IRF5s that we created, and I'll say more about that in a minute. Then there's an internal ribosomal entry site that allows other ribosomes to come in and transcribe ZS-green. So in both cases, whether it's the empty vector or our gene vector, delivery vector, or IRF5 vector, you have ZS-green as your reporter. So you're going to hope that when you see ZS-green in the cells, that means you delivered your IRF5 also. And you can check for that, which we did. So again, the first thing you want to look for in these experiments is that it works. You want to check your engraftment, same as we did with the other group. And you want to check your transduction, because you have an extra step here. So here we have our Fc gamma R2b IRF5 knockout background with our empty vector, and the same background, Fc gamma R2b IRF5 knockout, but this time TG is going to be the IRF5, exonal copy of IRF5 that we put in there. And here we have, going up, this is expression of donor CD45-2. So the upper group in both is the, this is not staying lit, but the upper group in both is the percentage that are coming from the donor mouse, which again is about 90%. And then in the second panel down here, then we started looking at, okay, of that 90% that came from the donor, what percent of them are expressing the ZS-green marker? The reporter that tells us that our transgene is delivered and integrated. And we're getting, in this group, I mean, this is a little under what we got. We got about 20-30% in the empty vector, and we got anywhere from 20 to 50% in the IRF5 system. So this one shows you 13, and this is an example of 55% in the wild type. So we had both engraftment and we had transduction. The transduction levels were not as high as we had hoped, but they were there. So we kept going. One of the next things to check was, okay, we have ZS green, do we have IRF5? Is there a protein? And we were able to get an IRF5, a small amount of an IRF5 antibody that was specific, and test and say, okay, now in my cells that have ZS green, do I have IRF5? This is all from one mouse, and in this mouse, this left, I'm just going to point this over. On this left side here, we have endogenous cells, CD45-1, and on the right side there, you have donor cells, CD45-2, and breaking the graph up on top, you have ZS green positive, and below that, ZS green negative. So, try this one. Is that better? Yeah, it's over. So you can see the endogenous CD45-1 cells never actually saw a virus, so they're negative for the virus, and they are expressing IRF5 as they should be, and it's all down here, about 10% of them are expressing it with the antibody that we used. Over here on the donor side, you get the reverse. If they don't have ZS green, they don't have the virus integration, the transgene integration, I should say, sorry. And you don't see IRF5, but when they did have ZS green, they do have the transgene and they are expressing IRF5. So this was a very good sign that we're actually generating protein. And we also looked at this to look at the fluorescence intensity of the amount of IRF5 that was picked up to see if we were getting a similar amount of IRF5 in the transgene versus an endogenous IRF5. And it essentially was, the shape is about the same for the amount of IRF5 being expressed relative to, I mean, the amount of ZS green, IRF5 being expressed, I should say, I'm sorry, in the cells. It was a little bit in the transgene, I guess you have a little middle population here that's expressing a little higher than the middle in the endogenous cells, but it tailed off in about the same direction. So this was good and bad news. Mostly it was good news. We were happy that the expression profile was very similar to a normal endogenous profile. It did mean that we really don't think we got overexpression. And that was one of the things we had said we were hoping to do, because we put a promoter on this that was supposed to be ubiquitous and overexpress IRF5, and we thought, hey, maybe if we can get overexpression going, we can see if that causes more severe or worse disease. We didn't. We got about normal expression. So good for biological relevance of the model, bad for our attempt to overexpress and see if that made a difference. So we have some speculation, but we don't have time. We can do it in questions if anybody has them as to why that might have been the case. But that is what we have. Looking at whether or not this was another interesting correlation that came up in the data. That was overall ZS green expression in the cells that we were looking at. This is in the spleen. And then we started looking at individual cell types to see if they were all expressing our ZS green marker equally. And whether the amount of ZS green overall correlated with the amount of ZS green that was in individual cell types. And in B cells, it did. And it was direct. The more ZS green you had overall in the hepatocytes, the more ZS green you had, and presumably IRF5 you had in B cells. In T cells, it did not. No matter how much expression you got overall in the hepatocytes, the T cells stayed pretty flat. So again, this was interesting. This is something that warrants looking into more in the future. We don't know whether this means that the transgene expression was pushing cells toward becoming B cells, or it could mean that T cells have a way of downregulating IRF5 production that we didn't know about. The good side of this is that T cells don't generally produce that much IRF5 normally. There's some literature that says they do it in inflammatory states. But so we were a little concerned with our ubiquitous promoter that we'd have overexpression of T cells, and what would that mean? It doesn't look like we got it. It was pretty consistent with the endogenous levels. So these are all pluses for the relevance of the model. So within the phenotype, we're going to talk about the same markers, so I'll go through them a little faster. But splenomegaly, we saw same thing, the guys that got the empty virus construct, normal sized spleens, the guys that got the IRF5 construct, some variability. Overall, a little bit lower than in the bone marrow chimeras. And that may have been due, so we're going to see that with a couple of the parts of the phenotype, and we think that might have been due to the fact that it was only in about 20% of the cells on average. So the fact that not every cell was Fc gamma R2B knockout, not every cell was IRF5 expressing might be why some of the phenotype is a little bit less robust. But interestingly, it is still within the ranges of a previous paper that our lab had published for systemic knockouts and systemic protection. So on the lower side, but still within normal, within bounds for what you would call disease and non-disease. Same thing with the lymph nodes. Nice spread on the guys that got the IRF5 transgene, nothing on the guys that were protected. We did do some positive controls in this group. I didn't show it other than this slide, but there was one group, I said, most of these experiments were on the Fc gamma R2B IRF5 double knockout protected background so that you could see the effect of the empty vector or the transgene. There was one group where we took IRF5 wild type. So it's FC gamma R2B YAA wild type marrow, and put that in with the empty vector just to make sure that they got disease. And that was, again, just to make sure we hadn't changed anything with the transduction and isolation. So that's the picture, the first picture here. They had nice, robust ANAs. Everything was, and all their other phenotype matched. I have that data. They look very much like the AM1 bone marrow chimeras. And then you get to the guys that got our transgene IRF5 are expressing, and the ones that got the empty vector were negative. So here we have the same data in graph form again with the scores. And this time you had a little bit of 1 to 100. One guy was at 1 to 300 in the empty vector group. But again, that's within normal limits for our knockout paper. You do see that sometimes. And if you remember, I said before, 1 to 100 is clinically insignificant. So there's only one that was really had a positive ANA in the empty vector group. And in the transgene group, 2 guys, for whatever reason, we think this might be related to the amount of ZS-screen in B cells, which I'll show you in a moment. But 2 guys did not develop ANAs, but everybody else was elevated and moving in the correct direction. So, yeah, speaking of that, we saw a definite relationship between the amount of ZS-screen, which we think means the amount of IRF5, as long as everything's working right, which we have indicated from our presence of IRF5 with our IRF5 antibody. A relationship between that and ANA production. So that in B cells. So what we looked at here is in just the B cells, the amount of ZS-screen that they're expressing, as it climbed, their ANA score went up. So correlation is not causation. This has to be looked into more, but it was an interesting result and fit with a lot of the other things we've been thinking, because it seems to indicate that IRF5 production in B cells is particularly important, especially to the autoantibody generation. Tissue, same things we talked about before. This is the blinded scoring. You can see a nice tight group that got the empty vector, good spread of disease, a little bit lower. This was pretty consistent in this model. The phenotype was a little bit lower, but still within a positive phenotype and statistically significant. Looking at 2 of the transgene mice, this is an example. In the mice that got the IRF5 transgene, we had disease in the kidneys. It was a little bit less pronounced than in AM1, but you still ranged from a moderate to a more severe glomerulus phenotype. And crescents. So that brings us to the main 2 groups we talked about. Real quick, I'm going to talk about one other group we tried. There was one other thing we tried to do, and it didn't work that well. We created another version of IRF5. Now, I talked about earlier, IRF5 needs to be phosphorylated to open up and dimerize and go to the nucleus. So one of the sites that they think IRF5 has to be phosphorylated at is this serine position 445. And when that site's phosphorylated, it opens the IRF5 up, and then it can dimerize and translocate. So we had a version of IRF5 where we converted that serine to an alanine, so it couldn't be phosphorylated there. And the hope was that then, if we put that in and we saw protection, we could say, look, when IRF5 doesn't get phosphorylated, it doesn't get activated, and you get a protective phenotype. When we did that, initially, things were looking good, because the phenotypes were very similar to the IRF5 knockout. Oh, I guess switch the slide, sorry. To the IRF5 knockout group. This is going to be them in the green. So there's our gene that got the empty vector, and there's our gene guys that got the S445A mutation, and there's our guys that got the transgene. So, and then over here you have percentage crescents in the kidney. You got about the same thing. So the S445A group was looking like the empty vectors, that they were looking protected, and so we were like, yay! But there was a problem. We weren't seeing ZS-screen in them. Typical GS-screen expression for our empty vector, 18-20%. Typical for our transgene over there, about 40%. And then you see over there the S445As, we're seeing less than 2% ZS-screen. So that's a problem, because without the ZS-screen reporter, we didn't know if they're actually making this IRF5 S445A mutation. And since it's on an IRF5 knockout background, if they're not making the 445A, they're protected because they're just knockouts at that point. So we don't know what to do with this at this point, because you can't tell if they're not getting sick because of the mutation, or if they're not getting sick just because they don't have the transgene and they're on a regular knockout background. So one of the things we did test these guys late with that IRF5 antibody we got, and we found that in a couple of the mice from the last group, even though they had no ZS-screen expression, they tested positive for IRF5 in these mice that should have been knockouts if they didn't have the transgene. So it looks like they may very well be making the S445A IRF5 mutant, But at that point, we didn't have the samples from the previous mice anymore, and we only had a limited amount of this antibody, so we couldn't go back and test them, so we can't draw any definitive conclusions. So in the future, it looks like this might have been a problem with the ZS Green reporter just in this particular construct. We could try troubleshooting this construct and troubleshooting the reporter, sequencing the construct to see if there was a mutation in the reporter that caused the reporter to fail. We could try running it with the same group and running IRF5 antibody detection to show that it is producing the mutated protein. But I think they've chosen in the lab to go a different direction because recently they said the heck with that, and they got a line where they had CRISPR-Cas9 and they put in the S445 mutation directly into the line, and they're going to study that. And they won't have to worry about this system. So, but they'll be moving forward. But when I found that out, after all the work we put into the S445As, that was my reaction. But the rest of the aim was basically a success. So we felt that even though the phenotype was a little lower, maybe like I said, maybe because we had it in less cells than we thought, it was still within the limits of what we see in the systemics, and it was still statistically significant, so we felt our data supported rescue of the phenotype with the IRF5 transgene. So that brings us to our last part, which is the IROC4 study. So when we got these IROC4, the first thing we did was we got a line of IROC4 mice. So IROC4, you have to understand, is a kinase. It can act as a structural scaffold where it's there for other proteins to attach to it, and it doesn't have to phosphorylate them. It's just part of the cascade and part of the signaling pathway. And then it can also act as kinase, where it's phosphorylating things. So there was some question. There have been different studies where they've knocked it out completely, but when you knock it out completely, you tend to lose NF-kappa B signaling also, because it seems to have a structural function in the NF-kappa B pathway. So more specifically, there have been studies where you knock out just the kinase activity, and you still get NF-kappa B signaling, but you lose the stuff that's dependent on the kinase side. And that's what we wanted to go after, since we wanted to see if phosphorylation of IRF5 might be IROC4 dependent. So we got a line of mice from Japan where there were mutations in the IROC4 kinase. There were 2 point mutations in the kinase that rendered it unable to phosphorylate other proteins. Otherwise, it was structurally the same. They had shown in papers that it could still fulfill its scaffold function. It just couldn't act as a kinase anymore. And we crossed that line into our Fc gamma R2B-YAA lupus prone model and back crossed it until the only mutation was the kinase dead mutation. And so these mice we're going to call Fc gamma R2B-YAA IROC4 kinase dead. Mouthful. So for the rest of this section, I'm probably just going to call it, I think I have them only referred to as IROC4 kinase dead, or IROC4KD. The other group that we're comparing them against is the Fc gamma R2B knockouts, Fc gamma R2B-YAA knockouts that get disease. And I'll call them, for the rest of this, IROC4 wild type. So just remember, everything's on the Fc gamma R2B-YAA background, but we're looking at whether they have IROC4 kinase dead or IROC4 wild type. So the first thing we did was a survival study to see if having the kinase inhibited, IROC4 kinase inhibition gave them a survival advantage in this model. And oh yes, it did, dramatically. The red line is our Fc gamma R2B wild type, IROC4 wild type mice, our regular lupus prone mice. You can see that they're all dead by 38 weeks, and their average lifespan was 27 weeks. The blue line is the guys that had IROC4 kinase dead. At the end of we had set a maximum limit on the study of 1 year. At the end of 1 year, not a single one of the mice had died. So that was a profound difference in survivability. When we looked at their spleens, massive differences. This is a good picture of what a Fc gamma R2B spleen looks like from the wild type IROC4 mice, and anywhere from 500 to a little over a gram. And then over here on the right, from the same model but with IROC4 kinase dead, you had actually slightly under normal sized spleens. Typical spleens, the data ranges anywhere from 80 to 100. These guys were in the 60s, so if anything, their spleens were a little smaller than normal. Same, that was the spleen in some pictures. The same here, in graphs. We actually had 2 groups we ran. We ran the 1 year survival study, and then we ran an additional 6-month group. And the reason we did that, we felt that the mice that were in the 1 year survival study, if we started comparing their kidneys at the end of a year to these R2 kidneys that had died at 6 months, that wasn't a fair comparison, because by a year they're going to start to have age-related changes in the kidneys. So for the year group, we basically focused on splenomegaly, lymphadenopathy, and proteinuria, but we didn't really look at renal histology. When we set a separate group up, we ran the separate group for 6 months, and at the end we euthanized everybody in both groups, and that's where we did our kidney histology comparisons. So you'll see there's a lot more spleens than anything else, because the first thing I noticed when I did that, I looked at the spleens from the 1-year group and the spleens from the 6-month group in the IRAK4 kinase deads, and there was no difference. So I combined them because there is no difference in a year. So there really was absolutely zero difference between them. As you can see, they're all straight across the line. There was never any splenomegaly in the IRAK4 kinase dead group. Whereas in the wild types, you get that whole range that's typical of the phenotype. Same thing in lymph nodes. This time, we chose to use proteinuria as a marker, which is when you have kidney damage, damage to the kidney, eventually albumin is a protein that's in your blood. When your kidneys are damaged, it starts leaking into the urine, you can detect it. And this is a semi-logarithmic scale, so every 2 steps in the scale is a 10-fold increase. And you can see the Fc gamma R2B-YAA mice have severe proteinuria, and these guys are actually from the 1-year study. At the end of the year, they had only +1, which is very mild, probably age-associated proteinuria, and that was statistically significant. We also looked at it in terms of, again, similar to the ANAs, we said you had to be higher than a +1 to be clinically significant, and a little over about 2/3 of the wild type group developed it, and only 2 of the protected guys are even there. And again, this was probably age-related in that group. Histology, you've seen this a couple of times by now. Guys, wildcat guys, very diseased. Guys who had IRAK4 kinase inhibition, very protected. There's a nice healthy glomerulus there. Same data, just this is our glomerular injury scoring. So again, nice disease in one group and nice basic protection in the other one, if minimal changes, if anything, in the protected group. So the last thing we wanted to do at that point, we thought we had established at this point that IRAK4 kinase activity was required for the lupus phenotype in our model. So the last thing we wanted to try to do was connect it to IRF5 phosphorylation. And the first way that we tried to do that was through a translocation study. I thought, hey, why don't we look and see if we can see the IRF5 activating and moving to the nucleus or not. And if IRAK4 kinase is required for that activation, then we won't see that in those mice. So Ying in our lab helped me out with that study, and the first thing we wanted to do was see if we decided to look at it in terms of, for this whole phosphorylation IRF5 study, we decided to look at it in terms of TLR7 responses, since TLR7, as we've seen throughout this presentation, is an important driver of SLE in the phenotype. So we said, okay, we'll take them, we'll stimulate TLR7, and we'll see if IRF5 goes to the nucleus. Unfortunately, we tried it in the wild type group first to see if we could detect it, and it wasn't really conclusive. It didn't really work that well. So that was my face there after that time. But we had another idea, and we said, all right, why don't we just look directly at phosphorylation of IRF5? And Alex Pellerin from our lab has experience working with phosphotag gels and had done it before and had the supplies and said he would run the gels for us. So he did, and thank you, Alex, because this was a fantastic result. This is my last figure, and then we'll get to acknowledgements. But this is an important one, so I'll walk you through. This is a normal gel on the left. This is a phosphotag gel on the right. And you have here splenic B cells that were isolated from either the Fc gamma R2B or ROC4 wild type, or the splenic or the Fc gamma R2B kinase dead. And then as a control group, he also took Fc gamma R2B ROC4 knockout. IRF5 knockout. Misspoke. I'm sorry. So yes, so we have everybody's on a Fc gamma R2B YAA background, and you have one group that was IRF5 knockout, and that's going to be our negative control to make sure that there's no IRF5 and that we're detecting IRF5 with the antibodies we used. Then you had another group where these other 2 groups, IRF5 is normal, but in one of them IRF4 is wild type, and in the other one IRF4 is our kinase dead version. So that's it, as I just said. Okay, so he also left some of them unstimulated. He stimulated some of them with TLR7 for us, and then in the last group he did TLR7 plus some co-stimulatory molecules to see if anything would get a response. And so let's talk about the gels. So IRF5 is this line here, and this is the IRF5 knockout mouse, and it produced no IRF5. and this is beta-actin as a loading control to show that we're working here. So everybody had IRF5 except the IRF5 knockout, and it was all at the same level. Okay, and then you go to the phosphatag gel, and in the phosphatag gel, there's an additive in the media that slows down the passage of phosphorylated proteins. So if anything is phosphorylated, it's going to run slower than the unphosphorylated version. And so when you look at it here, again, you got no IR5 in the knockout group. You've got the regular unphosphorylated IR5 in just about everybody. In the unstimulated, that's all you have. But then this is RH4/8 stimulation. And RH4/8 stimulation in the ROCK4 kinase wild type produces these additional bands, which is phosphorylated IR5. In the ROCK4 kinase dead, you get nothing. And then these are the same things as the other conditions, the RH4/8 plus the other co-stimulatory molecules. So across the board, you saw that whenever you stimulated TLR7 with or without the other molecules, in the ROCK4 kinase dead, you have no phosphorylation of IR5. So that was excellent, and the result we had hoped for. It opens up a couple of possibilities. It means probably either a ROCK4, it's possible a ROCK4 could be directly phosphorylating IR5, but it's also possible that it's phosphorylating something else, which is then phosphorylating IR5 in a chain. But it does put it in the same pathway, and it puts it as required. And so also because we're using TLR7 stimulation, we feel that this puts TLR7, the ROCK4 kinase activity, and IR5 pretty definitively in the same pathway. So that was good news. So that made me happy. And there's actually a TLR7 IR5 pathway there with the rainbow. So that was our third thing, and the data was supportive of that. And with the data being supportive of all three, we felt the data supports our overall conclusion. We believe that ROCK4 kinase activity and IR5 activation are both required in hematopoietic cells for development of SLE pathogenesis. So I don't want to put any pressure on my committee. They're free to do whatever they want, but I met Dr. Richard Dawkins last month, and he already said congratulations. Just throw that out there. Obviously, this whole thing's in memory of my parents. My father died right after my calls. He would have loved this. He was the first one in the family to get a master's, so it would have been cool for him to see me do this. But and then unfortunately my mother died last week, but they were inspirations. I wouldn't be here without the background they provided. So this is one of my favorite cartoons I wanted to share. Science requires long hours in the lab. My family hasn't seen me in a month. So one of the jokes we like to say is that in Homo sapiens there's a wild type gene for the life, and they're all having a nice time, and then people who have the knockout of the life gene are in the lab. My wife's here. She's amazing. I wouldn't be here without her. I'll tell her more later, but from the beginning when I decided to do this, we were talking about going back to school, and did I want to be a PA? Did I want to go to med school? Did I want to do grad school? And she said, whatever you want to do. And she supported it. So wouldn't be here without her. The kids and the dog you saw before. I'm glad they still know what I look like after the last month, so that's good. The dog's always great because she's the only one that'll come downstairs and say hi at 2 a.m. when I get up. My brother's here. Wouldn't have gotten through any of this without him as well. When my father died, my mother got sick, we had to divide up a lot of the responsibilities. I took on a lot of the healthcare decisions and how they should be treated, what they should be treated with, when to do DNRs and things like that. He took on all the financials. So he has handled all of their estate and the house and selling the house and trying to set them up and make sure that my mother was cared for after my father's death. And it was an unbelievable amount of stress and time on him and his family. I couldn't have done it. If it had fallen on me, I don't know if I would have finished. So he's a big part of why I'm here too. My cousin Melissa, who was always more like a sister, is here also. Again, Kevin lives up here in Boston, came here on a tour, and he got me interested in, I said, I'm looking to study APS, and he said, well, we study lupus, and we do these cool things with kidneys, and showed me some of it, and I said, that's what I want to do. I went and I saw him, and he said, well, I don't have an opening, but there's this guy Ramon across the office over there who has an R01, and maybe you could go work with him. And from the first time I chatted with Ramon, he was open to taking me on as a student. Yao is our technician in our lab who never says no to anything and is a great help and great support. Abraham's a former member of our lab who was a MD, but we did some experiments together. Kay, I can't say enough about Kay. Kay, our lab, we have MD PhDs. They have to split a lot of their time in clinic. Kay is our full-time PhD who is always in the lab every day. So she was the person that no matter what, I was there and she could encourage me or tell me, listen to me vent or like get feedback over my experiments or how they were going, and making sure that even when nobody else is around, that I was still on track and moving forward. So she's been invaluable. Ian, I talked about from the time I met. He's always been supportive. Gustavo, who's on my committee also, actually gave the lab the constructs that we use to deliver the transgenes. Prachi was an undergrad who's now in grad school doing a master's. She was a molecular biology specialist. She actually developed the construct with the different inserts for us that we used. Ying is a postdoc in our lab. I told you about her role with the translocation study that we tried to do. Alex is a PhD candidate in our lab who helped with the phosphatag experiment at the end. Amanda is a PhD, graduated before me, one of the first people I met here when I started, and we built on a lot of her work. She did some of the work in the knockouts and some of the first Chimera work that we had done in the lab. And Maggie Baker, who just graduated from our lab, who was always around to share helpful STEM reagents and other things that we needed. My wonderful committee, Matt, Ramon, Wayne, Gustavo, Jeff, who have been very patient and understanding and incredibly flexible with deadlines. And I appreciate all their support and advice over the time and their flexibility especially. Shamita, my GPG advisor, who has believed in me from the time I got here, from structure and function of the genome, and I still remember there was a test in structure and function of the genome that most of the class failed, and I had a ridiculously high score, and you guys were, that class, that was a rough one. That was a rough one. They recovered. I think you were sending a message on studying more for them. I don't know, but they didn't do so well, and I did. And as you gave it back to me, you kind of whispered, "You're like, good job." And Greg Vigilanti was right behind me. He goes, "I wouldn't have told you that," and he kept walking. But Shamita's always been there and supportive. Emily from GPGG has been great. She came in as our new kind of administrator for GPGG. She set up a camera for me today. She's responsible for all the food we had. She's been doing wonderful outings for our program, making us more social. Barbara Schreiber was my advisor my first year, for my full first year here, my academic advisor, and she stayed like a second mom or grandmother here for us. I'm very glad she was able to make it today. Andy Henderson's here today. Andy was my second lab rotation, which again made a huge difference. You've been there for me a few times, but I won't say who because I'm not disparaging anybody today. My first rotation didn't go as well. I was very new. I didn't have a lot of experience, and I had to learn a lot of techniques from the beginning. And at the end of it, my first rotation, my first advisor said, "Well, you're smart enough and you do really good research and you can look stuff up with the best of them and you're learning, but with a family and 2 kids and everything else going on in your life, I just don't see how you're going to finish." And so I was like, "Whoa, okay, thanks. Well, I'll show you, I'm going to finish." But then luckily, fortunately, my second rotation was in Andy's lab, and it was night and day. Andy had a family himself also, and understood and talked to me about what I would need to do differently and how it could be handled differently, how it could be done. His work at HIV was fascinating. He helped me learn to stay on task and balance life. And he's been there since then, too. Before my quals, when I was nervous, right before my qual exam, am I going to pass my qual exam? Am I going to pass my qual exam? I ran into Andy outside of the medical building, and he said, "I'm going to tell you something. When I took my qual exam, I knew the most about my topic that I ever knew in the rest of my life." And you will never have had the time to focus on this one thing as much as you did coming up to this moment. You probably know more than half your committee on this particular little niche topic. You'll be fine. And that gave me confidence, and I felt better going into it. And that and other conversations have meant a lot to me, so I'm glad you're here too. Thank you. And Dr. Borkin in the back there. Dr. Borkin filled in. One of my committee members couldn't be there for my qual exam, so he graciously filled in for my qual exam for that person. He was on the proposal committee, and he's always been there in the lab as a cheerleader and positive influence, especially late at night too, so I've appreciated all your support as well. And then my family we already talked about. My fellow PIDS and GPG students, my extended family, the Animal Science Center, for all their work with the mice that they did. I probably drove them crazy because I was very particular, but they worked with me. I think they liked it because it was very organized. But, the flow cytometry core, where we worked with, and the flow sorting core, which was invaluable to our experiments. And of course, funding. Graduate Medical Sciences provided us with a lot of funding when we had trouble with different R01s and other grants that kept us going and kept me able to finish. I will always be grateful for that. I had a 2-year ITP fellowship at one point supported by the immunology training program here, and that support was invaluable. And of course, Ramon's original R01. So thank you, everybody.